An antioxidant-rich diet, particularly one modeled after traditional patterns like the Mediterranean diet, has emerged as one of the most promising lifestyle strategies for preserving telomere length and promoting healthy aging. Telomeres, the protective DNA caps at the ends of chromosomes, are highly vulnerable to oxidative stress and chronic inflammation, two factors directly influenced by diet quality. A growing body of scientific evidence now supports the idea that nutrient-dense, plant-heavy diets not only enhance general health, but may actively slow telomere shortening by improving the body's redox balance and reducing cellular wear and tear over time. Fruits, vegetables, nuts, seeds, legumes, whole grains, and healthy fats like extra virgin olive oil form the foundation of antioxidant-rich dietary patterns. These foods are loaded with vitamins such as C and E, minerals like selenium and zinc, flavonoids, carotenoids, and polyphenol, all of which play crucial roles in neutralizing free radicals, the unstable molecules that cause oxidative damage to DNA. Telomeres, composed of guanine-rich sequences, are particularly susceptible to oxidative injury, which accelerates their attrition during normal cell replication. By buffering oxidative stress, a diet rich in antioxidants provides a line of defense at the genomic level, helping to preserve telomere structure and function. The Mediterranean diet, widely studied for its association with longevity, cardiovascular health, and reduced cancer risk, has shown notable correlations with longer telomere length in various population studies. For example, a 2014 study published in the BMJ found that greater adherence to the Mediterranean diet was associated with longer telomeres in a cohort of healthy women. The researchers concluded that this dietary pattern might act at the molecular level to slow biological aging. While observational, these findings are supported by strong mechanistic insights into how dietary antioxidants mitigate the pathways that drive telomere shortening, specifically inflammation, oxidative stress, and metabolic dysfunction. One of the most compelling aspects of antioxidant-rich diets is their systemic anti-inflammatory effect. Chronic low-grade inflammation, often driven by poor diet, obesity, or metabolic syndrome, contributes significantly to telomere erosion. Pro-inflammatory cytokines like TNF-alpha and IL-6 promote oxidative stress and interfere with telomerase, the enzyme responsible for maintaining telomere length in certain cells. Antioxidant-rich foods, especially those containing polyphenol like quercetin, found in onions and apples, resveratrol in red grapes and berries, and catechins in green tea, help suppress these inflammatory pathways. This leads to a more stable cellular environment that favors telomere maintenance and healthy cell division. Moreover, these diets tend to improve blood lipid profiles, insulin sensitivity, and vascular health, all of which are indirectly tied to telomere biology. Elevated blood glucose and poor lipid metabolism create a pro-oxidative, pro-inflammatory internal state that damages vascular tissue and promotes biological aging. By stabilizing these metabolic markers, antioxidant-rich diets reduce the systemic stress that accelerates telomere shortening. In essence, what is beneficial for the heart and blood vessels is often also beneficial for chromosomes. Importantly, antioxidant nutrients don't act in isolation. They work synergistically. The whole food matrix provided by fruits, vegetables, and whole grains offers a complex array of phytochemicals that interact with each other and with human metabolism in ways that isolated supplements often cannot replicate. For example, the combination of vitamin C with flavonoids improves absorption and recycling of antioxidants, amplifying the protective effect on DNA. This complexity helps explain why whole dietary patterns tend to outperform individual nutrient supplementation in clinical studies. Unlike pharmaceutical interventions, which can carry risks or side effects, antioxidant-rich diets present virtually no downside and many added benefits beyond telomere preservation. They reduce the risk of nearly all major chronic diseases, heart disease, diabetes, neurodegeneration, and even certain cancers. They also promote better mood, energy, and gut health. This broad-spectrum advantage makes them a cornerstone of any longevity-focused lifestyle. From a practical standpoint, transitioning to an antioxidant-rich diet does not require drastic changes. Even modest increases in fruit and vegetable intake can begin to shift the body's oxidative balance in a favorable direction. Swapping refined grains for whole grains, using olive oil instead of butter or margarine, and incorporating more legumes and herbs are all manageable strategies that contribute to better cellular protection. Berries, leafy greens, colorful vegetables, green tea, turmeric, and dark chocolate in moderation are among the most antioxidant-rich and accessible options available. In summary, an antioxidant-rich diet represents a deeply effective and evidence-based strategy to slow the pace of biological aging by defending telomeres from the inside out. It supports genomic integrity, reduces systemic inflammation, and fosters an internal environment where cells can function efficiently for longer. While aging cannot be stopped, the rate at which we experience it, both externally and at the level of our DNA, is profoundly influenced by what we eat. In choosing foods that nourish and protect, we send a powerful message to our cells: preserve, repair, and thrive.